is Wooster. And Luke has been doing some work with his research REU at Harvard. And he's going to talk about some of his results. The title of his presentation is Development of an Inline Microfluidic Electromechanical Cell to Study Carbon Dioxide Reduction.
My name is Luke Wooster, and I'll be recapping the research I've been doing the last 8 months or so at the Rowland Institute at Harvard with the Ferzoco group. So I'll start with the rationale for my lab's interest in an electrolytic cell. So my lab is a mass spectrometry lab, so my project is out of the sort of mainstream of their work, but it's necessary because the carbon dioxide 6-electron reduction to methanol is of particular interest to our lab. The reaction is of general interest because it converts a waste product back into a fuel. But my lab has interest because in the mechanism of methyl biologen's catalysis, there's a proposed step that's a proton-coupled electron transfer. And this is a rare thing to find in a mechanism, and we're hoping that downline in the instrument we can actually observe and characterize the proton-coupled electron transfer if it occurs. It's an unconfirmed mechanism that was proposed in a paper 2 years ago, and so we're working towards confirming it and then hopefully characterizing it. So here I have a quick diagram of what my cell will do in simple terms. Methyl biologen is at its most stable in its 2+ form, but that form does not catalyze carbon dioxide's reduction. And so my cell is needed to kick an electron onto methyl biologen, put it into its radical form, and that radical form is the active catalyst. And so once it's reduced, it can drive this reaction that we want to study. So this is a diagram of the entire proposed mechanism. You'll see there are 6 electron transfers, and I've highlighted several of the species that we can, sort of unique species that we can identify to sort of track the reaction as it progresses and confirm which route it took. So the top route, or only the bottom route, leads to methanol. And I don't have a box highlighting it, but you can see that at that 3rd electron step we have that proton-coupled electron transfer. Yeah, it's probably easier to do it that way. So this is a diagram of how my cell fits into the grander instrument that we're constructing. So my cell is in blue, and it sits just in front of the electrospray source for the rest of the instrument. It's hooked up to a potentiostat, which will read sort of all of the vitals, all of the electrochemistry going on. And then we source in the methyl biologen, and very importantly, we can tee in the carbon dioxide either before or after the cell. And this allows us to discern whether or not the catalysis occurs in situ in solution, or if it in some way needs to interact with the electrode surface. And that is also up for debate. The initial paper claimed that it occurs in situ, and several papers since have criticized that and sort of called that into question. So we'll hopefully be able to discern that by teeing in carbon dioxide before or after. So as we sort of embarked on this design, we sat down and fleshed out a few design parameters that we wanted to hold to. We needed the cell to play nicely with the mass spectrometer, so we needed it to have a flow rate that was compatible with the electrospray source. We needed as little dead volume as possible because we're generating intermediates that are unstable and will degrade quickly. And we ideally want a high sort of conversion efficiency. We don't know how much sample loss we'll have downline in the instrument, so the more we generate on the front end, the better. And so this is a basic sort of diagram of what the cell should look like. And this is where we started to iron out some of the dimensions of the actual channels of the cell. And so we were limited first by that flow rate. And so the flow rate and the length of the cell determines the residence time of an ion in the channel. And to get conversion efficiency, we want all of the ions in the channel to at some point encounter the working electrode. And so we need the height of the channel to be short enough that the diffusion distance across the height is shorter than the height of the channel. And so this led us to some sort of extreme parameters. We need the height of the channel to be about 10 microns, which is about 1/10 of a millimeter. So it's a very, very thin channel. And then a length of 3 centimeters would suffice for proposed 100% conversion efficiency though. And so going forward, we went with the simplest design we could because we were already playing with some really, really small dimensions. And so we just have a straightforward working electrode, and this again is where the methyl viologen will be reduced. So there will be a metal electrode there that will be a source of electrons for the methyl viologen. And as in any electrolytic cell, we needed a counter electrode where a sacrificial ion could be oxidized to provide those electrons to complete the circuit. And so we had some early tries that didn't work so well. We tried to sort of make a makeshift solution with an inline cell for HPLC, and that didn't work well. And then we tried some really crude sort of designs in the first few weeks just to see if we could do it with cheap materials that we had around the lab. And sadly, these failed terribly. So we moved to a microfluidic technique, which took more sort of work, but should work more reliably. And so this was very new to me, and I'm sure it will be new to you. It's a burgeoning sort of field that started about 30 years ago and has really progressed. And so now making microfluidic devices on the scale of microns is actually sort of easy once you get the hang of it. So you take a silicon wafer and you pattern a photoresist onto it called SU8. So SU8 is a positive photoresist, so wherever it sees light, it will polymerize, sort of harden, and you'll be left with a structure. And so all of this work is done in a clean room where everything is orange, so there's no UV light, and so you can very selectively expose parts of your SU8 to that UV, and you'll get to pattern structures very precisely. And so once patterned, you're left with your silicon wafer and then a mold of your channel structure. So the patterning is actually sort of simple in that you actually print off a transparency slide, and then that you can sort of make it in Photoshop and then print it off, and then that serves as the areas that will see the UV light when you expose it. And so this is a picture of the patterned silicon wafer. And so you can see the channel features are now raised up in SU8. So then we cast effectively a rubber over this. It's sort of a clear rubber called PDMS, but it's particularly useful because later on we'll be able to bond it very tightly to glass. I'll talk about that later. But for now, we mix up PDMS, pour it over the mold, and then we cure it and harden it. And then you can literally peel the PDMS off and then eventually bond it to glass. The bonding to glass is really nice. You expose it to oxygen plasma, and then you'll actually get a molecular bond between the PDMS and the glass. And so it forms very tight seals at the edge of the channels. This is a rapid prototype we did in the first few weeks just to confirm that we had a complete SU8 sort of mold and to confirm that we could get flow through all the channels. So you see again the working channel across the top and the counter channel below it. And it was food dye just to see it easily that we could flow through. So the electrode patterning comes next. It follows a very similar sort of pattern. This time we use a negative photoresist called Shipley 2012. And so it's the exact opposite. Wherever it sees UV, it will soften. And so we expose it there so that it disappears in the middle. And then we do E-beam evaporation of titanium and then platinum to actually deposit metal on the glass slide. After that's done, we can expose it, or dissolve it really, in acetone. This is a picture right before we dissolve. So that's right after it's come out of the deposition chamber. And so platinum's just been applied. You can see the yellow underneath is the photoresist. So after we expose to acetone, all of the photoresist is washed away. And so all of the platinum on top of that photoresist goes with it. And so we're left with just platinum where we've patterned it, where the photoresist is initially exposed. And so this forms sort of the other half of the cell. So we have our PDMS, and now we have a patterned glass slide. And like I said, PDMS and glass can bond very tightly. So we sort of slap them together, and we have a completed cell with a channel then down. So this is a picture of our actual cell hooked up to another mass spec that we have in the lab. It's upside down, which is a little tough, but that's sort of the easiest way to orient it for actually flowing through. But you can imagine that flipped. It should look familiar from the design I showed at the beginning. This may look innocuous, but this was an exciting moment for the lab. This was the first droplet of solvent that we pushed through. So this confirmed flow through to the electrospray source. That's the sort of nozzle for the instrument. And this is initial data to confirm flow through. And so this peak here is methyl viologen registered by the mass spec. So we've really flowed methyl viologen all the way through the cell and then recorded it with our mass spectrometer. And we're not using it electrolytically yet, and so we're not seeing much methyl viologen 1+ generated, but we're not expecting to. Now, before we identify electrolytic activity with our mass spectrometer, we wanted to identify it with the potentiostat. And so we did some initial cyclic photonograms just to confirm that we're able to reduce and oxidize. And so this is methyl viologen being reduced and oxidized, and this is ferrocyanide being reduced and oxidized. It's sort of a really nice PET chemical to test a lot with. It's really stable, and its redox properties are well understood. And so this is just a quick summary then. My cell reduces methyl viologen to 1+4. It sits right in front of the rest of the instrument, and we're hoping soon to generate these intermediates so that we can study them down the line in the mass spectrometer. This is not our instrument. This is sort of another mass spec we have in the lab just to do testing on. Actually, I have a picture here. This is our instrument. So this is our baby that we work on. So that's where I'll leave it and open it up to questions.
Questions for Luke?
Like all that stainless steel there, it stays nice and shiny, clean. The proposed system uses methyl viologen as a catalyst. Is there enough known to know how stable that is over time, reusing the catalyst over time? Is there enough known at this time?
We know that it's not particularly stable, and that's one of the factors that drove us towards a microfluidic design. We wanted very, very little dead space. So the actual residence time in the channel is about a second and a sixth, like 1.6 or 1.06 seconds. And then its residence time after the channel to the mass spec is even shorter. And so we only have like about 10 microliters of dead space. And so we're hoping there's not enough time for it to degrade back or, you know, throw its electron off to something else and go back to MP2+. But it isn't super stable. So we have to deal with it. I have other pictures. This is the instrument that does the deposition. And this is where, like I said, everything's orange, so there's no UV light until you want there to be. And this is where the SU8 and S2012 casting is done. And then this is what we have to wear all the time. It's sort of a chore, but we have to wear these in the clean room to keep sort of dust and anything out of the air. Since features are so small, dust particles sort of look ginormous to your cell. So everything's got super good.
We're actually ahead of schedule.
We are. Thank you, Luke. If I could put in a shameless plug for history, I would, but I can't. I really can't at this point. Thank you, Pierre Richard, for taking over this last session. I appreciate it. Well, what a couple of few hours we've had. Last night, many of you were present when Sean Coleman talked to a more general audience than is here this afternoon and said, get in the game. I think what we've seen in the past several hours is that there are a number of ENC students across the disciplines who are in the game. And it's inspiring. Luke, I can't tell you what you're talking about. I told my colleague Ben not to ask any questions. I'm sure he doesn't either. But that's not the point. The point is that in a variety of areas, ENC students, and encouraged and modeled by ENC faculty, are in the game. And that is so encouraging when you're at a small school that maybe you think is sort of underneath the radar in terms of American higher education. And yet there are people here doing wonderful things and have been doing wonderful things for decades and will continue to do so, all ultimately for the glory of God. It's an inspiring thing. Thank you for all of your work. There are others who were shy or reticent to bring forward their own work. I hope maybe next year they'll be willing to do so. Again, I say it's inspiring. I thank all of you for participating, and I thank all of you for attending. I believe, Sarah, that much of this, if we can, we'll work on the details, but it's been videotaped, and we'll try to put this on the honors program website so that it can be accessed both by our own people and people from outside. So this won't be hidden under a bushel, but will be there for other people to see. Again, it's been an honor and a privilege. Thank you for coming, and we'll rack them up again next year. Take care.